Okay, so welcome all of you again for second part of session 7. In this session, we are continuing to talk about the GPCR receptor and G protein signaling. In this session, we will talk about the effector, the mechanism of the effector activation and second messengers. And at the end of this lecture, we will give you some clinical examples of various biological response that we could get from GPCR and G protein activation. So, the aim of this lecture, we will understand how we could get activation of the effectors like cyclic AMP or specific ion channel, and later on to get a specific cellular response. And we will give you also some examples to explain to you how GPCR and G protein signaling is significant for various physiological processes. So, receptor activation could lead to the generation of a signal directly. This is true for those signaling molecules or those ligands which are lipid soluble, like steroid. They are going to act on target protein inside the cell for the steroid one, such as they are going to act on a specific receptor within the nucleus that lead to the transcription of various proteins, or water soluble ligand that bind to a receptor, the receptor located on the ligand gated ion channel that lead to the influx of specific ions that lead to the direct physiological response. However, the vast majority of our biological response require receptor and then effector and second messenger. So the first effector that we are going to talk about it is the adenyl cyclase. In previous session, we talked about how activation of the G-alpha S, for example, by binding of adrenaline into the beta-1 adrenergic receptor in the heart, lead to the activation of the G-alpha S protein, and this one lead to the activation of adenyl cyclase enzyme. That's a specific enzyme embedded on the plasma membrane. During resting state is inactive. After activation of G-alpha S by binding of noradrenaline to the GPCR, beta-1 adrenergic receptor, and exchange of GTP for GDP, and G-alpha S GTP go to act on adenyl cyclase on the plasma membrane, and activation of adenyl cyclase lead to the hydrolysis of ATP to cyclic AMP, and cyclic AMP later on go to act on second messenger, which is the PKA, protein kinase A. Such adenyl cyclase could be inhibited by G-alpha I, not G-alpha S. S for stimulatory, I for inhibitory. So when we have, for example, specific ligand bind to a GPCR, like for example what you have alpha 2 adrenergic receptors that bind to the adrenaline or noradrenaline ligand, lead to the activation of the G-alpha I protein, and this one lead to the inhibition of adenyl cyclase enzyme, and later on inhibition of the protein kinase A, and inhibition of various biological response that we could get from PKA. So this slide will show you the steps in getting second messenger and specific biological response. Starting from the beginning, we require ligand. This ligand need to be water soluble in order to bind to the GPCR receptor. Such binding between the ligand and GPCR lead to the activation of the G protein. If the activation of the G protein is G-alpha S, lead to the activation of adenyl cyclase enzyme in the plasma membrane. Activation of adenyl cyclase enzyme lead to the hydrolysis of ATP to cyclic AMP, which is the second messenger. The first messenger here is a water-soluble ligand outside. The second messenger is a cyclic AMP. And cyclic AMP later on go to act on protein kinase A enzyme. And protein kinase A, the major action of these kinases is to phosphorylate other proteins or ion channel to get a specific biological action. This image interprets the previous slide in an image. For example, we have a plasma membrane, we have a GPCR receptor. GPCR receptor bind to the first messenger, water-soluble ligand. This one lead to the shape change of the G protein. The G protein, as we explained, consists of alpha subunit, beta subunit, gamma subunit. And alpha subunit, inactive state, bound to GDP. When we got a ligand bind to the GPCR, GTP will exchange for GDP in order to get activation of G alpha GTP. Such G alpha GTP, if depending on the receptor and ligand, if it is activating G alpha S go to act on this enzyme, which is already embedded on the plasma membrane, called adenyl cyclase enzyme. And adenyl cyclase enzyme lead to the hydrolysis of ATP to cyclic AMP, and cyclic AMP activate protein kinase A, and protein kinase A go to phosphorylate various ion channel and proteins to get a specific biological response. Protein kinase A has 4 subunits, 2 regulatory and 2 catalytic subunits. Cyclic AMP go to bind to the regulatory subunit to release the catalytic subunit. And the catalytic subunit later on active and go to phosphorylate other ion channel and proteins. So that is the first effectors and the first second messenger pathway that we could get from GPCRG protein activation, which is a cyclic AMP, adenyl cyclase, protein kinase A, and please don't forget this first effector. So generally, what will be the action of adenyl cyclase and cyclic AMP and pKa on our physiological response? Usually, to understand or to have an idea generally about various physiological action, I am always asking you to remember the sympathetic and parasympathetic nervous system activation. For sympathetic nervous system activation, always we are looking to fight and flight response. And parasympathetic, we are looking for rest and digest response. For fight and flight response, we require increased heart rate, increased cardiac output, increased blood pressure, increased muscle contraction, and all of this require a lot of energy, which we could get it from in terms of the glucose. And this fight and flight response, we will get it from adrenaline or noradrenaline ligand that bind to specific GPCR receptor, alpha 1 adrenergic receptor, or beta 1 or beta 2 adrenergic receptor. This coupling between adrenaline, noradrenaline, and GPCR receptor lead to the activation of the G alpha S or G alpha I, and after that we will get specific biological response. So if I require energy, I need to break down storage glycogen, I need to break down storage lipid. So this one is a lipolysis, and the former one was glycogenolysis, and we need to get gluconeogenesis within the liver. Okay, and I need to increase the heart rate by acting on beta 1 adrenergic receptor that lead to the activation of G alpha S and then pKa, and pKa act on the L-type calcium channel to influx more calcium inside, and to get more cardiac heart rate. When they increase, they will increase cardiac output. When cardiac output increase due to the increased blood pressure. So the cardiac output, when they increased, it will be required to provide enough blood supply for skeletal muscle contraction that contain enough amount of oxygen. So fight and flight response could summarize to you the action of cyclic AMP. And these are some examples of the physiological or biological actions that we could get from this effector enzyme and second messenger. Second effector enzyme will be phospholipase C. In calcium signaling session 5, we talked briefly about phospholipase C. Now we will extend this explanation a bit more. So we have phospholipase C enzyme embedded on the plasma membrane during resting state. When this enzyme becomes activated, you will get cleavage or hydrolysis of PIP2, phosphatidylinositol bisphosphonate. When PIP2 hydrolyzes or cleaved, it gives you IP3 and DAG. IP3 by itself go to act on IP3 receptor on sarcoplasmic reticulum or endoplasmic reticulum. The binding between IP3 and IP3 receptor lead to the release of the calcium from the storage plate, which is ASR. Such calcium release go to act on calmodulin, and calcium calmodulin complex go to act on various protein and kinases. But how I could get activation of the phospholipase C enzyme? I need G-alpha-Q GTP activated. Such G-alpha-Q GTP protein, in order to become activated and dissociated from beta-gamma subunit, it requires binding of ligand to GPCR receptor. For example, binding of acetylcholine to GPCR muscarinic receptor type 3 lead to the activation of the G-alpha-Q, and G-alpha-Q is always activating phospholipase C. And phospholipase C will become activated, lead to the degradation of the PIP2 into IP3 and DAG. DAG, diacylglycerol, activate protein kinase C, which induce transcription of various proteins within the nucleus. And IP3 go to bind to the IP3 receptor on the membrane of SR, and then release of the calcium. And the calcium act on various protein kinases by binding to the calmodulin to make calcium calmodulin complex. So initially we have a water-soluble ligand like acetylcholine bind to the GPCR receptor like muscarinic receptor. This one leads to the activation of the G-alpha-Q, and G-alpha-Q GTP activate phospholipase C enzyme embedded on the plasma membrane. The activation of the phospholipase C enzyme lead to the hydrolysis or cleavage of the PIP2 into IP3 and DAG, and IP3 by itself lead to the release of the calcium, and calcium will be a second messenger to act on various protein and kinases after binding to the calmodulin to make calcium calmodulin complex. In order to explain more in image this information, so we have G protein coupled receptor. Such G protein coupled receptor bind to a first messenger, which is a water-soluble ligand. If this messenger and G protein coupled receptor are specific for activation of the G-alpha-Q, such G-alpha-Q lead to the activation of the phospholipase C enzyme, and phospholipase C enzyme lead to the cleavage of the PIP2 into DAG, diacylglycerol, and IP3, methyl triphosphate, and IP3 then come to bind to the IP3 receptor on the membrane of SR that stores the calcium ions, lead to the opening of this channel and release of the calcium, and calcium bind to the calmodulin and act on various proteins and kinases to get various biological responses. So phospholipase C is actually a very large number of the G protein coupled receptor could activate this pathway. For example, as we explained, acetylcholine via muscarinic receptor, histamine, noradrenaline via alpha-1 adrenal receptor. So alpha-1 adrenal receptor is actually located on smooth muscle and vascular smooth muscle. Binding of noradrenaline or adrenaline to the alpha-1 adrenal receptor lead to the constriction via G-alpha-Q activation and calcium release. So we need to always remember that phospholipase C enzyme activation will be via G-alpha-Q. Please don't forget this one. Activation of adenyl cyclase will be via G-alpha S. Inhibition of the adenyl cyclase will be via G-alpha I. Please don't forget this information. And after that, we will get various effects from such activation process. So the common mechanism in signal transduction is a protein phosphorylation. So you are putting a phosphate on various proteins or enzymes, specifically on specific residue on the amino acid or the protein or enzyme named serine or serine. Putting phosphate on these protein kinases or enzyme lead to the activation of these kinases or enzymes. And after that, you'll get specific biological action. In this table, we summarized some of the protein kinases that could be activated by specific messengers, such as we explained, cyclic AMP will activate protein kinase A, PKC by DAG, PKG by cyclic GMP, calcium commodity complex after releasing of the calcium from the storage place within the SR. And each protein kinase causes phosphorylation of various other target proteins like enzyme, ion channel transporter, cytoskeletal proteins, which could give you various biological action. So one of the characteristic features for any signal transduction is amplification. Amplification, you are enhancing the signal. You are making your signal very strong in step by step. This process is very important in order to get a required biological action. For example, you have ligand like noradrenaline or adrenaline bind to beta-1 adrenergic receptor, which is a GPCR receptor. This binding between the GPCR and noradrenaline will not activate only one G-alpha S. Will activate many, hundreds of G-alpha S. And each G-alpha S will activate not only single adenyl cyclase, will activate hundreds of adenyl cyclase. And each adenyl cyclase will not activate or will not hydrolyze single ATP to cyclic AMP. Will hydrolyze thousands of ATP to cyclic AMP. And each single cyclic AMP will not only hydrolyze single pKa. So this process is called amplification, that we are required to get a proper signal transduction. So in each steps of the signal transduction, we need to amplify this signal. So such amplification could be in the beginning, when single receptor could activate more than one G protein, and each activated single G protein could activate multiple effector molecules, not only single. That's the second amplification. Third amplification, for example, activation of adenyl cyclase, which is an effector enzyme, could hydrolyze thousands of ATP to cyclic AMP. So you note that such amplification at different steps. In this picture, we could see that single GPCR activation could activate more than single G protein, and this one could activate many effector enzymes, and the effector enzyme could hydrolyze many ATP, not single one, and this could activate many protein kinase A. So if you take a look at the first one, which is the one ligand, however, at the end, how much you could get? Thousands. That's the amplification. So termination of the signal. We need to go back to the basal state in order to be ready for a second message. This process of deactivation could be performed by many ways. For example, we explained GTPase enzyme activity that hydrolyze GTP back to phosphate and GDP. The GDP bind to alpha subunit and GDP alpha subunit rebind to the beta and gamma complex and will become inactivated. So GTPase enzyme activity could be accelerated by various kinases inside the cell. This one of the way to switch off the activated G alpha protein. Second way, the binding between the ligand outside and the GPCR by time will reduce the binding. The binding will become more weak and more weak by time. So you will get dissociation. So one of the way could be receptor binding to the ligand lose such strength by time, or could the receptor itself become phosphorylated. So I need to explain this point a bit. So when GPCR bind to the ligand become activated. When become activated, lead to the activation of G alpha subunit and G beta gamma subunit. G beta gamma subunit could recycle recruiting some proteins called G protein receptor kinase. These kinases go to act on GPCR, lead to the phosphorylation of GPCR itself. Phosphorylation of the GPCR will be a target to bind to another protein called arrestin protein. Binding between phosphorylated GPCR and arrestin will trigger internalization of the receptor. This is another way to remove the receptor from the membrane via internalization through the endostic vesicles and recycling the receptor back to the membrane. This is another way of the deactivation. You could read more about this process of internalization of the receptor, and I'm not sure if you got it in your previous session. But this is the internalization, one of the way to desensitize the receptor. So the first one, less strong connection between ligand and receptor. Second one, internalization of the receptor after being phosphorylated. Third one, by enhancing the GTPase activity. By enhancing GTPase activity, you are hydrolyzing the GTP back to the GDP and phosphate. And this one lead to the bring back G protein into an active state. Or we have some enzymatic activities within the cell. For example, protein kinase A could activate phosphodiesterase enzyme. Phosphodiesterase enzyme, what is doing? Hydrolyzing the cyclic AMP to 5-AMP. 5-AMP is an active state. So pKa could act as a negative feedback on its own by activation of phosphodiesterase enzyme. Or inositol triphosphate that lead to the release of the calcium from the SR could be inactivated by inositol phosphatase enzyme. So we have some enzyme inside the cell that could inactivate second messenger and bring back the cell into the basal state. And also we have some other enzymatic activity that could not only act on the second messenger, could act also on the targeted protein kinase. For example, in smooth muscle contraction, when you have calcium bind to the calmodulin, such complex lead to the activation of the myosin light chain kinase. The myosin light chain kinase after that will phosphorylate the head of the myosin, and this will lead to the cross bridge formation between actin and myosin, and contraction and shortening of the muscle. Myosin light chain kinase could be inhibited by myosin phosphatase. So you have some enzymes, which is, for example, myosin phosphatase, could inhibit the already activated target kinase. And our example was myosin light chain kinase. So in musculoskeletal module, you will get also such pathway, this pathway of the small muscle contraction. So it could be receptor ligand being not binding anymore. That's one of the way. Second way could be by internalization of the receptor. Third way, by increasing GTPase activity. Fourth way, by some enzymes activity, like degradation of inositol triphosphate biphosphatase, or inhibition of cyclic AMP by degradation of the ATP into 5-AMP biphosphodiesterase, or by final way, activation of some enzymes that lead to the inhibition of targeted kinase. All of this will lead to the deactivation of the or switching of the signal, transduction.